Hello everyone, my name is Yang. I am a PhD student graduated from UCLA Bioinformatics program, and I'm currently working at Children's Hospital of Philadelphia with my PhD advisor, Dr. Yixin. So in today's talk, I'll be introducing a tool that we recently published to study tumor transcriptomes, especially alternate splicing alterations during cancer progression. I'll be talking about the computational frameworks of the tool and also some biological insights we gained by applying the tool. A quick background. As we all know, alternative splicing is a very prevalent mechanism of RNA processing that exists in nearly all human genes. It gives rise to diverse types of RNA and protein isoforms. What I show you on the right is basically different types of alternative splicing patterns, including the simple form like exon skipping, alternative splice sites, intron retention, and so on. It can be a little bit more complicated, like the complex alternative splicing patterns I show you at the bottom. So this process is actually being tightly regulated, and the dysregulation of it is a major cause or contributors to human diseases, including cancers. In fact, aberrant isoform expression is widespread in tumor cells. A recent TCGA pan-cancer paper showed that there's an elevated level of alternate splicing in cancer comparing to normal samples, which can be up to 30%. Well, it's not only about numbers, but also about functional significance. Alternative splicing has been reported to occur in genes in every category of cancer hallmarks. As shown in this diagram below, there are alternate splicing involved in the gene called CD45, which is related to avoiding immune destruction. Similarly, alternate splicing has been reported in genes that are involving evading growth suppressors, resisting to cell death, and so on. And this is simply based on a review back in 2013. Well, with so many large-scale RNA-seq studies during the past 7 years, we can only expect the number of evidence to grow. Over the past decade, the community has witnessed an exponential growth of sequencing data. As example, TCGA, the Cancer Genome Atlas, have generated more than 11,000 patient-derived tumor samples, covering more than 30 different tumor types. And similarly, we have GTAC that sequenced another 10,000 samples derived from normal tissues, covering a wide range of tissue types. Along with other large-scale sequencing efforts, they together presented an unprecedentedly exciting opportunity for knowledge discovery, especially for studying the regulation and dysregulation of transcriptome using large-scale RNA-seq datasets. And fueled by the rapid growth of sequencing data, advanced algorithms have also been developed to leverage a variety type of sequencing data. For example, a recent pan-cancer analysis backed by TCGA have reported hundreds of thousands of alternate splicing events being detected in large-scale tumor RNA-seq datasets. In other words, it's not uncommon to detect hundreds of thousands of splicing alterations in large-scale tumor RNA-seq datasets. However, how this large amount of splicing alterations are regulated during tumor development remains unclear. So in order to address this issue, we collaborated with the VT Lab and the Black Lab from UCLA, and we established a pathway-guided approach to better answering this question. The specific type of cancer that we're interested in is prostate cancer. So this cancer starts from normal epithelial and evolves into adenocarcinoma, and this is often referred as a primary prostate cancer, which is relatively benign. However, it can progress into metastatic prostate cancer, including the castration-resistant phenotype and also the neuroendocrine phenotype. So this metastatic prostate cancer are often with poor prognosis and very deadly. To study the entire disease spectrum of prostate cancer, we compiled a comprehensive dataset with nearly 900 samples from different sources to represent different stages of the disease. Through a uniform RNA-seq processing framework, including using RMAT, which is a tool that we developed in our own lab, we identified hundreds of thousands of alternate splicing events in this dataset. So for the rest of my talk, I'll be just focusing on the most common simple form, which is called the skipped exon events, as you can find in the table on the right, is in the first row. I think it's worth to mention how RMAS and many other splicing quantification tools to quantify alternate splicing. It basically uses this metric called psi value or percent spliced in, as I typed below. It's nothing but a ratio between reads that supporting the inclusion of the middle exon and the short reads that supporting the skipping of the middle exon plus the inclusion of the middle exon. So it's a ratio between 0 and 1. 0 means a completely skipping of the middle exon. 1 means a total inclusion. So after proper filtering of coverage of the junction and also other filters, we're able to end up with more than 13,000 splicing events with high confidence. This allows us to define the splicing landscape of prostate cancer. As I show you on the right is a PCA plot based on these 13,000 splicing events. It can group samples by their cancer phenotypes regardless of the data source of origin. By comparison, on the left, a similar analysis using the same dataset but use isoform level count-based metric, we cannot see that. We can see that the samples are grouped more by the data source instead of the disease stages. This suggests that ratio-based splicing metric can be more robust and mitigate the batch effect, partially due to the ratio-based metric being self-normalized by comparing two forms from one event. And the result also confirmed that we can robustly capture biological signals from the splicing data. But we're still facing the problem that there are over 13,000 splicing events. How are they being regulated in tumor during the cancer development? In collaboration with John, we were first inspired by decades of cancer genomic studies, where genomic signatures were established to reflect the disease stages and oncogenic signaling activation. So we thought to leverage those gene signatures to guide our search, we were able to identify splicing variants responding to oncogenic signaling alterations during the development of cancer. So following this idea, we implement PEGASAS. As you can see in this diagram, PEGASAS starts by taking RNA-seq data from patients' tissues across different stages of the disease. And by taking the same RNA-seq data from the sample, PEGASAS branch into two orthogonal measurements, pathway activity and alternate splicing. So for pathway activity, PEGASAS leverages the ranked gene impression and KS statistic to come up a pathway activity score. So for splicing, we leverage RMS to quantify psi value for each event. And this together would allow us to perform a correlation analysis with a permutation procedure to get rid of the false positives. And at the end, we're able to identify pathway-related splicing events in a given tumor RNA-seq dataset. So the next thing we did is actually to benchmark the pathway activity score calculated by PEGASAS. We utilize the MYC oncogenic signatures as the most established and validated one. We tested on the primary prostate cancer from TCGA as it has rich annotation of MYC activation, like a copy number amplification or mRNA overexpression. As a result, as you can see from the box plot on the left, MYC pathway score can identify samples with MYC activation either through MYC genomic amplification or MYC gene overexpression. And we can distinguish those samples from those samples without MYC activation, for example, the benign primary prostate cancer and the primary prostate cancer. And we can further stratify patients as we show on the right. Using MYC pathway scores, we're able to stratify patient survival, showing comparable performance with MYC copy number signatures, which is considered as a hallmark of MYC activation. And notably, MYC pathway score can help to determine samples of MYC activation even without seeing any genomic signatures or MYC gene overexpression, highlighting the advantage of using gene signature set instead of using single gene measurements. Then we carry out PEGASAS analysis on 13,000 splicing events from the prostate cancer dataset. We identify the set of splicing events associated with MYC activation Among them, I just want to show you two of our best examples in terms of correlation coefficient. So on the left is this splicing factor SRSF3, but also it's actually a proto-oncogene. So the exon inclusion level or step value on y-axis is strongly anti-correlated with the MYC pathway activity score on x-axis. Looking at the disease stage through this sashimi plot, we can see that the middle exon shows a steady decrease from normal to metastatic phenotype, despite a rebound in neuroendocrine phenotype, which is actually from a relatively small sample size. On the right, we saw a similar trend of MYC-correlated exon skipping event in this proto-oncogene called HRAS. Expanding the PEGASAS analysis from MYC pathway to all other curated hallmarks signatures maintained by the ISCB. As a result, I show you this heat map where each row represents one pathway, each column represents one splicing event. Pathways are sorted by the number of splicing events associated with. We can see that E2F, G2M, and we can see MYC among the top. Looking at the functional organizations, we found that the top gene ontology terms of those splicing events are mostly RNA binding, indicating oncogenic pathway correlated splicing enriched in RNA binding proteins. Next, we selected 7 oncogenic pathways that are known to be associated with prostate cancer. Again, we saw E2F, MYC, and mTOR are among the top in terms of number of splicing events that are associated with. It is more or less expected that these powerful transcription factors, like MYC, could strongly impact the transcriptome landscape. However, what is really interesting is that when we're looking at the genes that are harboring those splicing alterations, they are predominantly coming from spliceosome-related genes, suggesting splicing machinery itself is a major target of oncogene-driven alternate splicing, and it's potentially functioning through a self-regulatory mechanism. Well, this is a very interesting phenomenon, but we all know that the correlation doesn't suggest a causation. So far, everything is based on large-scale correlational analysis. Experimental perturbations are needed. Therefore, John Phillips from the VT lab developed and engineered a human prostate cell line with regulated MYC expression. Specifically, the cell line derived from a normal human prostate cell with an enforced MYC overexpression controlled by the drug doxycycline. So the withdrawal of doxycycline will reduce the MYC expression dramatically. So in the paper, I didn't show the data here, but we extensively validated the cell line and to make sure that it resembles the MYC-activated prostate cancer. So here I will skip all those details and we'll be just focusing on what we can learn from this cell line. So what can we learn from the developed cell line? We sequenced 3 biological replicates of the engineered cell lines under 2 conditions, MYC overexpression and withdrawal. By performing a paired test, as I show you here, we're able to identify over 1,900 MYC-dependent splicing events. Similar to correlational analysis from the patient samples, we saw that the MYC-dependent splicing events are strongly enriched in genes affecting RNA splicing-related processes. Intersecting this set of events with the previously identified MYC-correlated events from patient-derived samples, we're able to see a significant overlap with 147 shared events. Among these events, we're able to validate our top examples, as I show here, SRSF3 and HRAS. So for both events, we observed MYC-driven exon skipping from our cell line MYC withdrawal experiment. So what's in common in these 2 events is that the middle exon both contains a stop codon. So for HRAS, the stop codon will lead to a truncated isoform, which named as P19 can act as a suppressor of tumor growth. For SRSF3, the stop codon will act as a premature termination codon, or PTC. So the inclusion of this poisoning exon would result in nonsense-mediated decay, leading to the decrease of the corresponding protein level. So similar case can be actually found in more than 1/3 of the MYC-regulated events. You can see more details from the paper. And we also found another 1/3 with potential to introduce the frame shift that will cause the inclusion of the points in exon. So this together suggests that MYC may play roles in splicing dysregulation of PTC-containing exons. So what may be happening here is that using this SRSF3 gene as an example, so exon number 4 of it contains the premature stop codon. The skipping form of this event will lead to the normal translation of SRSF3 protein. When the protein expression level goes too high, it will auto-regulate and bounce back to the exon number 4, which will induce the inclusion of the middle exon. And the inclusion form will trigger the nonsense-mediated decay, which will eventually lead to the decrease of SRSF3 protein level. So this ISMB loop is actually not a new mechanism. It has been discovered for more than decades ago that exists in many SR protein families. And it also has been shown to be evolutionarily conserved. But what we found novel here is that it seems the MYC activation may allow the escape from this autoregulatory mechanism and eventually achieve a dysregulated level of SRSF3 protein expression level in prostate cancer. This actually were experimentally confirmed by the immunoblot data from our MYC cell line. We saw that the SRSF3 protein level decreased relative to the housekeeping gene GAPDH under the MYC withdrawal. So the very last question we asked in this analysis is that, is this a prostate cancer specific phenomenon or is more generic? We expand this analysis to 2 other human epithelial malignancies. We found that there's a conserved set of splicing events associated with the MYC oncogenic activation. So one of the ongoing work is actually to investigate the interplay between oncogenic pathway and the splicing in a pan-cancer scale. So I just summarized my talk here. We developed the PEGASAS, a computational tool to study alternate splicing alterations during cancer progression. And by applying PEGASAS to study prostate cancer, we established a rule of MYC in regulating RNA processing by controlling the incorporation of points in the exons. And also I want to point out that PEGASAS is actually a generative framework to study other type of RNA processing dysregulations other than alternate splicing, and it can be applied to a pan-cancer level analysis. With that, I want to thank my lab members, collaborators, and funding agencies. And thank you for your attention. I'm happy to take any questions.